Good evening, Dr. Kasala. Can you hear me?
Yeah, I can hear you.
Okay, good. So, shall we start the session?
Yes, we can start now.
Yes, please start the session. Before that, those who are present here, kindly let us know whether you have understood yesterday's topic, whether you are clear with the scripts which has been shared. Have you worked on that? Please give us a thumbs up. Please let us know whether you have used the script as much as you can. You are okay with the scripts. Is there anything you want to say us regarding that? Your feedback is most welcome now. If you have understood the script and you have worked on that, you have got the output, please give thumbs up. Or if you are yet to work, let us know that as well. Before starting, let me know what's happening in you. Okay, Divakar, do you want us to assist on the error? Thank you, Imran. Ghazala, meantime, you can start sharing your screen. I'm waiting for you.
Yeah, I will share.
Yes. And Mr. Divakar, do you want us to assist on the error? I mean, Dr. Ghazala want to see what is the error. Do you want our guidance? Okay, Divakar, thank you. Others? I have got two reactions. Is Rathava. Right, one. I need to practice again. No problem. You can take your time. If you have any doubts, you can make use of Telegram. Others? We have got response from three. It will be good if others also give your response. Please let us know what's happening with you. It will be good. I don't want all to give the negative feedback at the end. I mean, what's happening, we just want to know then and there so that we will try to rectify it. This 15 days is only for you.
Okay, so do we have
That,
queries? Shall I check the check
no,
box
no, we don't have
first?
any queries. We are just discussing about yesterday's concepts. And it seems Lavanya has got an error. Miss Lavanya, do you want any assistance from Kasana?
Yes, please.
Yes. Could you please say the error?
It says something like I'm able to get the program in I mean script in the box, but I'm unable to run it when I hit run. But it's saying something. Vintages contain introductory material. Bioconductor is a package installed, but I'm not unable to run the program, the script.
Okay. Can you please share your
Okay.
screen? We can rectify it.
So
Is it okay for you now?
maybe after class, I don't want to
Okay.
interrupt other
Okay.
people.
Okay.
Yeah.
Dr.
Yeah.
Ghazala, is that okay?
Yeah. Later, like in Telegram, they can share their error screen,
Okay.
like a
Okay.
screenshot of that, so that I can check that because
Yes.
already we
If
have
time
less time.
permits, we can have after the session, maybe Ms. Lavanya can share the screen. But if we extend this time, sorry to say, Ms. Lavanya, you can post your error in the Telegram that we can
Sure.
work on.
Sure.
Yes. And
Okay.
those who are, all others have said they are working on the scripts. They have yet to complete the script. No problem. You can try your level best to complete. If you have any questions, you can make use of Telegram forum. Thank you guys for your response. And Dr. Kasana, please start the session.
Yeah, we'll start the session now. Okay, so today we will be using machine learning algorithms to design the model based on our gene expression data. Let's just have some basic concepts like we know now by you already know from where we are getting the data and at which step we are going to implement machine learning algorithms to make a model for our data. And basic knowledge you already know, like what is difference between traditional programming or even statistics, and difference between machine learning, that also we discussed earlier. So we need some data, and when data is not classified, or even sometimes we have labels in our data and sometimes we have no labels, so for that also we need some data cleaning option. Or like yesterday in gene expression analysis, we itself saw, like we use principal component analysis to reduce the dimension and visualize the data in one frame, so that we can see that which data or which sample is outlier in our data, which may lead to misguiding results. So that we use for data cleaning in, so that type of approach we also use in machine learning for data cleaning. Again, we have two types of, like broadly two types, but we have multiple categories. Like we have also, along with supervised learning techniques and unsupervised learning techniques, we have reinforcement learning technique also. But usually we use these two techniques for our data when we are having like less complicated data, like only textual data we have, or we have only image based data. And again, that depends on which model we are selecting, that depends on what type of data you are having, either it is labeled or not labeled, and what is your objective. So this is just a brief overview that we have already gone through, that we are going to use different types of algorithm now, because we are using gene expression data and we have all our data labeled, because we know which samples belongs to healthy samples and the disease samples. Also, we have the gene information, all gene IDs we are having, because we have annotated our data after normalization. So we are going to use that data for analysis. So we will be using supervised algorithms uh, to develop a model for our data. So again, we have the text form data right now for training and testing, and then we have labels in our data, so that will go into supervised learning. And the what goes at the back end of the algorithm, that you have we have discussed like in brief how algorithms actually work, but this is not for all algorithms. Different algorithms, different process. There is a certain calculation going with the algorithms at the back end that you don't see. You just have to give your input, and you will get the the required output will be given by your model, which is developed based on your data, or which has learned from your data. Okay, so this is the basic pipeline for machine learning analysis, where you have the data and you will go for data preparation, and your raw data will be normalized first, or you can also, so you can either use your raw expression, like the gene expression that we saw in geo database. We downloaded the matrix file, right? So there in the matrix file we had the raw expression of all the FEIDs, or the probe IDs for each corresponding gene. So you can use the raw expression as well as the normalized data that we have. And after cleaning, you will only proceed for training and testing, splitting of your data in training and testing set, and build a model based on that. So this will we will follow, and different classification techniques we have in this list. Some of them are not listed here, but we can add more if if we require. If our the and these algorithms are not working on our data, or not giving enough accuracy based on our data, then we can also use different. So not we are not going to perform the analysis with all these tools. Few of them we will use, because we have less time. And then again, here we have a distribution of the algorithms based on what is the objective of your analysis. Whether you have the numerical data and you want numerical output, then you will go for regression. And then if you have classification, like categorical data, like we have healthy and disease samples, so that is a category. We have two groups, And then there we will be using classification algorithms where logistic regression, KNN, SVM, random forest, and all. So other types of algorithms are decision tree, XGBoost, and categorical boosting techniques. So these are based on ensemble methods because it will be using different approaches to vote for each. Like in decision tree, we make trees based on different number of leaves or root nodes that we saw in decision tree, and we will see in few minutes. And then based on the voting from each approach, what is the prediction identified by that tree at the end, that will be taken for the final result. Also in random forest, since it is a collection of decision tree, so it will again get prediction from each tree which is included in the random forest, and then it will give the result. So similarly, like that, all the ensemble algorithms or the methods are depending on different types of algorithms used in it, and then give the result. Again, then deep learning is since the subclass of machine learning. So deep learning, okay, some of you has queried that deep learning is not discussed. So deep learning is used because we are right now using text-based data for the gene expression. So we are not having as of now complex data, because deep learning algorithms we use when we have complex data. For example, like millions of rows and columns we have, or sometimes we have text image form of data, we have audio data, like audio recordings are there, and we are segregating the voices of say uh, like people, different people. So that type of data, or even if we have video recordings, so if we are segregating images from those videos, or collecting different images out of those videos, so that type of complex data, we use deep learning algorithms. So all depend on data that we are going to use. So this is just a general format of decision tree, how it will take decisions. So it will depend again on us, that how many number of nodes we are trying to give, and how many leaves will be generated. Leaves are the decisions made by the different conditions we are giving to each node, and whether the value is more than 10 or 5, whatever threshold value we are keeping for each decisions. So that will be taken by the decision tree. Again, random forest, a collection of decision tree. So it will work on the majority voting and averaging. That is the ensemble method, where different algorithms. So in ensemble method, what happens, not in random forest, only decision trees are taken. But in different, if we are using other ensemble method, or we are analyzing data with ensemble approach, we can also use different algorithms here, like decision tree. Then we can use SVM, and the output or the prediction identified by KNN, and then collection of those algorithms, we will identify the final model or the final result based on the majority voting. Then the KNN will make clusters for your data based on the type of pattern it is showing in the data. So if it can classify your data into 2 groups or 3 groups or more, based on what type of data you have, or the labels on your data. For example, if we are using 3 groups, we have healthy samples and the disease samples. So just the 2 groups, DCIS group, but there was one another group which was IDC, invasive ductal carcinoma, which we have not used for analysis. But if we have taken that, then we, in our KNN plot, we will be having 3 different clusters. So that type of clusters it will make. And then we have naive Bayes and other type of algorithms, logistic regression, so for use for categorical analysis, and then a support vector machine to again classify 2 different groups in our sample based on the pattern that shows within it. Okay, so neural network is based on DLL, like this also we briefly discussed in previous sessions, that it will work based on different hidden layers. So no more number of features in your data, more complex or more number of hidden layers will be there in your network. Okay, so these number of layers you will be deciding that how many number of layers you want, and as you increase the number of layers based on your data, the accuracy may also increase at the end. Okay, so that also depends on again data and the complex data that we have. Right now we have text based data, so we will be using not algorithms which are basic or traditional machine learning algorithms, because these type of algorithm which have too many number of layers, and they are like highly computational, they need high computation. Okay, so it will take some time for analysis also. And if we are not having complex data, we will not use this type of algorithms. We can use neural network, and I may also show you during analysis, but ultimately it will not give much better accuracy because our data is too general or text based, not too complex to analyze by traditional approaches, that can give better results in less time than using deep learning algorithms. So based on the complexity of data, we will choose the model that we are going to use. Again, choosing a model is one of the very important step that we are going to use. And someone has asked that why we are using different models or analyzing the data or making different models using algorithms. So we are just trying to explore different type of algorithms, and what we already know, the like when you go into the description of all the algorithms that are there in the machine learning uh, books, so you will see that for each algorithm the pros and cons are given, that some will work on more larger data set, some will work on less data, like shorter data set that we have just few columns or rows, 10 to 15. So, and sometimes some algorithms take more time and more computation power. So based on that, we will try to see like what computation power we have currently for data analysis. And then again, what type of data we have. For example, if you have categorical data, then you will try to use only those algorithms that are going, those are having categories.
Miss Anu, is there anything you want to say? Okay. Rasala, please go ahead. It seems by mistake.
Okay, so yeah, so selecting model is important because and you will select model based on the data first, you data that you have, and then based on the performance or the what is the pros or cons of the algorithm. Because if one algorithm need larger data set and you are providing just 5 different columns to a algorithm, it will not give good accuracy, or and it will not also be uh, like uh, useful for your analysis. So based on the pros and cons or the characteristics of the algorithm itself, how it works actually, and what is the data that you have, you need to select the algorithm. Right now, or for our analysis, we will be using different algorithms so that we can see how these different algorithms work on same type of data, and based on that we can also see along with that based on accuracy and all, that how it performs on the data that we have, because we know that we have 25,000 rows and like 19 columns as a feature. Okay, so quick review for the features. You already know that we have different columns that are related to the samples that we have, that those are columns, and then features are different samples that we have in our data. So healthy samples, and we have the group. Okay, so healthy and disease samples are made in two groups, and based on that the labels are given that whether the sample is healthy or the diseased. Okay, so then that was just an example. And then based on your features and the label that you have given, the algorithm will try to learn the pattern from it, that how it changes when there is a healthy sample, and how the expression of the gene changes when there is a disease sample. Based on that, it will develop a model for you so that you can use different data on the same model. Okay, so once the model is made already, you can go for model optimization, where you can try to tune different arguments that you are providing to your data. For example, in random forest, you can decide how many number of trees you are giving or you want to give, or you can change in each step, so that you can see that how changing of different decision trees varies and the accuracy of your algorithm. And even before before developing the model, you can also set for, like, just to avoid the bad performance of your model, you can also go first set the cross validation step, so that your model that is being developed is already trained, check the data 5 times and trained and learned the pattern 5 times by taking different sections of your data. So these sections, you will be deciding either you are going with 30% and 70% of your data for training or testing. So once you have already decided that how much data you want to provide for training and testing set, you will see how the model is being developed. Then we have model evaluation matrix, where we will be looking at the prediction of the model, that once the model is prepared, we need to see how it is predicting of working. Either it has learned enough on your data, or it needs more, like hyperparameter tuning, or even more data we can. So if we have less data, we also try to make simulation data for that, and make the data set larger, so to make the model learn more from your data. Okay, so we have 4 basic terms that we use in this confusion matrix, where you will be having true positive and true negatives. So that will be telling you. So that, and also the false positives, which are if we are taking positive as having disease, so false positive will mean that there is a disease, but the model is predicting falsely and saying that it is not a disease sample. That is called a type 1 error. And then false negative. So the model is saying there is disease because it is negative, but actually it was the disease sample. That is type 2 error. So this will make a confusion matrix like a tabular form, and it will tell you that how your model is performing on the data, or how it has learned from your data, based on what is the actual values and what is the predicted values on X and Y axis. Okay, so in this description, you can see how false positives, which were the type 1 error, says that healthy people, it has predicted healthy people incorrectly as the sick people, because these were the healthy people. And then the false negative has incorrectly. So negatives will be applied when the model is predicting wrongly. Okay, so that is type 1 and type 2 error as false positive and false negatives. You can go through the slides so that you will understand better. Again, the evaluation of the model will be made on based on accuracy, that how much accuracy it is providing for the prediction that is making. So there is a formula that how accuracy is calculated, and which is again based on the confusion matrix values. So summation of true positives and true negatives, and then which will be divided with summation of all the values that is there in the confusion matrix. that will be providing you the accuracy actually. So whatever accuracy you are looking into your output while doing writing programs or like analyzing your data, you will see based on this calculation only. So either you are looking at accuracy, or even if your confusion matrix, you can also calculate the accuracy and other metrics based on the confusion matrix if you have. So here, if you see the table, there is an example that we have true positives 30 and then some true negatives. When we calculate, the accuracy is 0.96, which looks the model is performing well, okay, on your data. Okay, so also apart from accuracy, there are other metrics, that is sensitivity, also called as true positive and recall, which can be calculated again based on the values in your confusion matrix, like true positive divided by true positive and false negatives. And then there is a specificity, which is measure of the proportion of actual negatives that are correctly identified. So that has the formula again based on confusion matrix, and you can calculate once you have the confusion matrix by yourself, or you can write the program, or if you are doing web-based analysis, then you can calculate by that. Recall is actually sensitivity. Here we have see that there are different terms we use for similar type of word or the estimation that is provided by the model. So recall is sensitivity, and this will tell you how many actual records are correctly predicted, or how many actual samples are correctly predicted. And then there is a precision, which is 1 minus specificity, and that which has different formula and will tell how many correct predictions out of all the predictions. So again, these are the evaluation metrics that you will be looking at in your data when you are trying to evaluate the model that you have just developed. Okay, so I think that is clear for all. If there is any query for anyone, they can ask. Okay, I don't think we have any query. Heat map. Okay, few of you had the confusion about heat map. So maybe by the end of this session, I can explain you. If we have some time, I can explain you about how to generate that heat map file. Actually, let me see if we can do it now. Okay, so this is actually post-normalization expression, and we have the probe IDs here, but we need the annotated post-normalization data because there we will be having the gene symbols as well. So let me see where we are having those files. Here we have, let me just close this first. Okay, I also added the VLOOKUP formula for you in the Telegram channel. So please check that so that it will be useful for you when you are looking at it, like you are using that. Okay, now I'm opening the filter DEGs that differentially express genes. Okay, so DEGs we use in short. I am using that. So here we can see we have total all the FEIDs which are not actually filtered. So we can first filter based on p-value. So first select the p-value column, go to your data, and then filter, apply number filters. So we will do this just real quick so that we can go for ML analysis. This is taking more time because the expression file is also open and it is big. Okay, so let me just show you just mapping, because that this step I think you all can do. Filter is a very basic step. Yeah, so now you can see here we have the table. Everything is going so slow, so just bear with that. We have just two files are opened, but why it is taking more time? Okay, so hope you all have generated this file. Like when we are generating the final result file after the EG analysis, we are getting without symbols column, without the gene symbols column, but you need to annotate this file using the annotation table that we are having. So what we will do is this table we are having the annotated symbols, and we will filter the genes. Right now what I am doing is I'm not going to filter because it is taking more time. So I will just try to take few genes from here, just selecting top 20 genes, adding in the next worksheet so that we can just see how we are mapping. So we need this column, symbols column, to map the gene IDs. We have some common genes you can see here with different FEIDs. So you know now how to filter these genes based on log FC or even p-value. So both ways I have shown you how to remove this duplicacy using the log4 change value filters or the p-value. So don't try to remove duplicacy using both p-value or log4 change value, but just use any one. I have shown you both, so that's up to you, or based on the target gene that you are looking for. Okay, now we have the gene symbols. We want to see the gene expression, the normalized expression of our genes for only these genes, like which are differentially expressed in our data. Like they seems important genes in our disease samples or in the development or progression of the disease, ductal carcinoma in situ. Okay, so what we will do first, we need to have this file opened and then go to your Excel, open the expression file. We have the symbol columns here. Just generate one new column before just after the symbol, like whichever column you want to map, generate a blank column. After that, write the formula, because first row is for the header, so that we are not going to use. We will go with the first gene. Okay, you write the formula. We look up, it will already come if you are starting with is equal to sign, and then when you write we look up, it will come appear there. Okay, you use that. Then once the bracket is open, select your gene, any the first gene you select, so that later it will select all the genes accordingly. Okay, so first gene you are selecting. Once you select, it will show you the B2 cell you are selecting in the Excel. Once it is selected, give comma for after your this cell number, and then open your Excel, the other Excel from which you want to map this gene, that you want to identify whether this gene is there in my DEG list or not, because we want only those genes which are differentially expressed, not all the genes. So we will go to our filter table. We will select the column which we want to map. We are mapping symbol column. So select the symbol column. Now it will appear here. The whole sheet name or the Excel file name will be here, or right now you have opened in CSV. So it is CSV file. Name will be here. Then sheet number 1, because you are using sheet number 1. And then there is a column, B column, which is total B column is selected by you to map the gene. Anywhere in the whole column, the gene ID is there, it will map. Then use 1 for exact map and then 0. If it is exact map, then give the number. Then close the bracket. Okay, so 10 you need to remember to get the exact map of your gene that is there in your DEG list. Now hit the enter and then you can see we got not available. It means not available. This DDR1 gene is not there in your DEG list. Now go at the end, this you might know that for the autofill, or you can just stay here on NA and use some option here. I think for autofill there is a option. Let me see. We have option in cells, or just let me show you. You can explore it later because we usually do. This is the shortcut that you can go at the end. There is a square box. You can just double click and it will fill the whole column based on the same formula that you are using. You don't need to write the formula again and again. It will use the same formula that you are using for the first gene. Now it is mapping the second gene to your next on the DEG Excel, third gene to your DEG Excel, because you can see as you move through the cells, it is changing the cell number, the B6. Now here in sixth row, it is using B6, okay? And in the fifth row, it is using B5. So it will change automatically if you are going with autofill. Once it is autofilled, you can see a lot of NAs are there. It means not available. So these genes are not available. What we can do first, every time you are doing VLOOKUP or using any formula, remember to paste your data whole column as a values. Okay, so select the whole column, Control C for copy, or you can use copy from here, Control C, and then paste here itself, or in any other column you can also paste. So let me just show you by pasting in any new column. Like by the end here, I am trying to paste as a value. So right click and in paste option. So for some of you, these might not be appearing due to version change in the Excel. So you can go for special pasting and there it will show you different type of options. Okay, so maybe there you will see the paste as values option. Here is the paste as values. Now once you paste, now when you go through cells, it will show NA directly, not the formula. But here when you are going with the raw formula columns, then it will show you formula. Okay, but we don't need formula. With formula, it will not work if you are moving this column anywhere else in your file. You can now delete this column because it was having formula, and have this column moved here. Now you have this mapped DEGs, so you can write it DEGs so that you will be knowing that these are DEGs that you have just mapped. Filter it now because we don't want not available genes. Filter it for all the genes. So you can see on filter, we can see how many number of genes we are already having in this list. But on top, it is showing all not available only. So we will filter and remove the filter for NA. Okay, so you can select from here in the box that what genes you don't want to see. I don't want to see not available genes. So select NA and give OK. Now you will see all the genes which are mapped. Okay, so copy this whole table or use Control A to copy, to select all and then paste in the new sheet so that you can see how many number of genes you are having as DEGs, and for that only you will now copy the whole table from here to the using DEGs and the all the samples that you have, and then use this table to make a new Excel file and save it, use for heat map. That's, I think that's clear for everyone. So use these DEGs to plot the heat map. Hope that is clear, and formula you already have that we were using. So use that. I'm closing now. I hope that is clear. That was just a revision you needed. Okay. So we will try to do this analysis first. For machine learning, we will be doing the analysis. So let's just open our tab. Okay, here in machine learning, what we need to do is first load, like as usual, while working with R Studio, you first need to load certain, install and load certain packages that is required for the functions that we are going to use. Okay, so I am loading XLS because I have data in my Excel file. Yeah, one more thing, you will be wondering that from where we are getting the data. So the data we are having in here, the same data. I have prepared a short table, but you can see the whole expression table. The normalized data that we have, I have used it. Like here, you can see my preview tab. So without opening the data, I can show you here. In the preview tab, we have symbols and all the samples that we have for healthy samples and the tumor samples we have. So this whole data I have in expression table that we use for used or generated after normalization. Okay, so this data we are having, and since it was having too many genes, like around 22,000, so what I have made a short table for that, so that we can quickly get the results from the algorithms. Otherwise the model will take time, like it will take some time to make the model, because data is more, then training time will be more, and then testing time will be more. So for that I have made a short table. Again, in short table, you can see there is a symbol, and then samples again are all the tumor samples and the disease and the healthy samples we have the annotated data, because we right now we can see we have symbols, that is the gene symbols that we have. Okay, so short table we are going to use. Let's see. After loading the packages, okay. After loading the packages, we can see here. So we need some certain packages. I have added some comments that why we are using this package. So read Excel is for reading the Excel file, data in Excel file. Tidyverse is for data manipulation and making different plots. Caret is using like Caret library is having most of the packages for, or the functions for machine learning analysis, like random forest and all type of algorithms that have. So basically all the traditional machine learning algorithms are stored for them. Functions are stored in Caret library. So this is must for your machine learning analysis. And GLMnet we are installing for this elastic net regression analysis, which is a logistic regression. Okay, so that we will be seeing that what type of algorithms we are using. These are common um, like commands, like graphics off to close all the plots that you are having in the day in your environment where you are working around now. And then RM list is to remove the whole workspace where you are working right now. Here at the bottom, whatever in the console you are having, or whatever packages you have already loaded, you will remove everything. And control at L you use whenever you need something to, like you want clear console. Okay, so use control L. See, with control L we can just clean the console, because uh, be because sometimes there are a lot of errors and like other output that we have generated for different programs. We don't need it. We want to start from fresh. So, okay, so we will start by loading the packages first. Okay, so here you can see I'm reading data from, not from CSV now. I'm using from short table and also Excel data. So, and my data is stored in sheet number 1, and sheet at position 1. You can also give sheet number. For example, your sheet number is sheet name is DEG. So you can also give DEG here, but right now my sheet, I am giving sheet number. So sheet number is 1, and that you already know, like how, what in Excel we call sheet and sheet numbers. Let me open my short table. So this is, I think, most of your confusions that from where we are getting this data for to analyze in machine learning. So this is my data and this is my sheet one. We can give it any name, but whenever we are getting the data, we can say this is expression data. Okay, so you can also write expression data if to retrieve this data or if for your sheet, or you can just give sheet number one. If it was in sheet two, then I would have given given the number as two. Okay, so these are the orders of sheet in Excel to get the data. And this is the same data that we are having. Yeah, so now I think, okay, we need to read the data first from Excel and store it in df variable, which is the data frame that we are getting. So run the data, and now once we have load the data already, now we will perform data pre-processing. We call it data pre-processing while we do gene expression analysis. But when we do machine learning, so what we do, we call it exploratory data analysis. Okay, there we will try to make certain plots so to visualize the data, how our data is looking at exactly. So what we do, we need to use certain codes to view the data first, like we need to see the head of the data. So head of the data that we have just retrieved, like here we have df. So df is for data frame that we are having, and we can see top few rows of our data using head. So this will show top 5 rows of your data, and here is the top 5 rows where you can see or the gene symbols are there, and then sample names are there. So these are not the renamed samples. I'm using that old file that we saw in the first day of the analysis, and we have the gene IDs and we have the gene expression. Okay, so this is just to view the data, then see like exactly what type of data format you have. Like you can see there is the first column with symbols, and they are using as character. Like you can see for each gene ID, which is or gene symbol that you are seeing, they are closed in double inverted quotes, and they are as the format CHR, which is character format within 1 to 100 characters in each data. So this is just to explore your data, because we are doing exploratory data analysis, which is just to explore your data, that how your data looks, what is the format of your data. And then again, you have columns like normal, all the healthy sample column is here, where numbers, they are in form of numbers. Then all the data after that, either it's a disease sample or either it is a tumor sample, it is in number format. Okay. Okay, so no, number format is basically numerical format that we are having. Now you can see the dimension of your data, that how many number of rows and columns you have. I have selected 100 genes and then 20 samples that we are having. So I think I have used more samples in that, or I have added more healthy samples because we had less healthy samples, only 5 samples. So I added some samples. Let me just see there so that you will have no confusion for that. Yeah, we have, you can see at the bottom in Excel, we have 20 samples. I think I have just simulated some tumor samples. We have 14 tumor samples. Yeah, 14 tumor samples and five five healthy samples. So 14 tumor samples are, some of the samples must be replicated just to make our data a little big, so that algorithms or the model that we are going to prepare should learn better with more data. Okay, you can also do with less sample, but in that case, less number of samples, but in that case, you need to carefully select your algorithm to train the model. Okay, so yeah, this is it. Like we have 14 tumor samples and 5 healthy samples. And first is your column that we have. So we have 100 genes right now. And then, so I already mentioned that I have made a short table. You can go with the like whole table also, but your model will take some time, so you need to just wait for the model to get trained. Okay. Yeah, so that's why we need GPU and sometime TPUs for working on bigger data. Dimension we have already, that we can see how many columns and samples we have. Now, if our samples, for example, if we have already not removed, not available, or the gene IDs that yesterday we were getting, some of the gene symbols were showing no NA for no gene ID mapping to that FEID or the probe ID. So if we want to remove with the like code, we can remove. If we want to remove in directly in Excel, we can filter the data with NA values and we can remove those rows also. So both ways we can do. So this is the code just to remove the NA values from your data, and then again you can visualize your data. So let's just see like what it is showing. I think we don't have any values right now, but still we can see if there is any change, because this head top 5 rows we have already seen. So it will show the same top 5 rows because we don't have any values as of now. If you have, you can remove. So this is because any values is removing. Here what we are doing, we are handling with the missing values. We can remove the columns directly, or we can put some alternate, like if the gene ID is not given, we can give gene one some name giving to that gene, so that later after analysis, if we find that gene important, then we can explore or annotate that gene or identify that what is the gene ID for that probe ID. Okay, so this is handling the missing one step of handling missing value by removing the data that is not having, or the rows that are missing having missing data. Then we will see that how many duplicate genes we have. uh, I am not sure if we have duplicate genes, but we will see with code. Okay, so we have both options. We can see these, like we can do these steps in Excel also, like by filtering the data to see the duplicates. Yesterday we saw by highlighting red color for duplicate genes and how to remove those duplicates. So we are doing that step here. Each step I can explain you, but it will take more time. That's why I'm just trying to skip directly to machine learning. This is exploratory data analysis, and this has too many steps, because you need to create or generate plots, different type of plot you can try, different type of, like right now we are looking at how many duplicate genes we have. So this is one of the step, but it is not necessary as of now, unless your model is not unless your model is not able to handle with duplicate genes. For example, some models may work with duplicate gene and they will consider that gene as a single gene, so or it will automatically remove those duplicate genes. So in that case you don't need these steps. So these are kind of optional steps which you can use or not use, totally depending upon you. So we have again 100 genes and 19 columns right now, because it is excluding the column that is having the gene IDs and just having the sample IDs. So we have 14 tumor samples and 5 healthy samples. That makes 19 total samples. Now here we are like doing some data manipulation here by converting row names as first column, and so first column we are having as a symbol name, like gene symbols that we are having. So renaming your sample and everything, so that also we can do. Sometimes when we are working with like writing code and doing all the analysis, so we don't want to go to Excel and do some analysis there. So we use different code for these type of small things, like renaming the column and all, which is very easy in Excel if we are doing just typing there the other name, but we are doing it by hands. like by writing code here rather than going to Excel and doing it manually. Now we want to transpose our data so that our genes goes on the like as a column, and then and all the samples. So here the whole table will be transposed. For example, let me show you with an example. This is the short data that I'm taking out of it just to explain you. So this is the table that we have, and we want to, let me give it a short name so that it will be easily useful for you. Okay, so this is the sample table we have. We want to transpose table so that all the genes goes like will convert to, rather than as row as a column, it will go in a first row, and then all the samples will come as a first column. Okay, so when we copy the, we can do it manually also, but why we are not doing, let me just show you. So here when we paste, go to special pasting, there is an option to for transpose. So we can also do this manually here. Like when we are pasting data as a transpose, now you can see all the samples are here and all the genes are here. So this we can do, but why we are not doing this step manually? First is because we are writing code now and we want to do everything with code because that is easier to stick to one interface when we are doing intensive data analysis. And second thing is that because right now we are having 101 genes, because I have just made a short data, but when you are using all the genes for your analysis and making the model, then you will be having 22,000 genes, and Excel does not support that number of columns, like 22,000 columns. Excel will not support. So for that reason, we cannot do that manual work. So we are doing with code. Okay. So that is the one step that we need, and we will try to save our transposed file in CSV so that we can use that transpose table later. So that is one thing. DF, okay. Did we just transformed it? No. So this is for the transpose table. Let's just transpose our table. Okay, now table is converted. When you will open this transpose table, we have already saved it like here, modified. So here is the transpose table, the first one. And when you will open it, you can see the samples on the in the first column and all the genes in the first row and the corresponding expression. Okay. So when we transpose, everything changes. So all the gene expression will, like if your gene, the second gene is moving to second column here, then its expression will show here only. Okay, so don't worry about changes in expression if we are transposing the table. It will, they all work together. So transpose will happen all together and gene corresponding expression will stay there with that gene corresponding to that sample. So don't worry about data. Again, once we have transposed the table, so we are reading the transpose table again to visualize the dimension, because now we have more rows and columns. Yeah, here, dimension. So this is for dimension. Yeah, now we can see, earlier we were seeing 100 samples, 100 genes first and then 19 samples later. But now we are seeing 19 samples first because now we have this in the first column, and then we have 101 columns because 100 genes, and first column is for sample names that we have as the healthy samples and the disease samples. Okay, so here, this one is just to view that what your first column look like. So your first column is having header as symbol, and all the healthy samples and the disease samples are in your first column. That's what we can see with this ID. Now this is just the data operation where we are trying to remove like replace your underscores. Like here you can see we can use g sub function to replace your underscore or remove all the information after underscore, because we have underscore in your data or the file name you can see, or the column name you have underscore and then all the information, then you can use this to just remove that. So different type of things you can do this manually in Excel, but I am just showing you because some of you are curious to see that how things happen with the code. So these are just basic data analysis or data manipulation things where we are just reading the data file in different variable. So the data frame we transpose. So T I have just added with the variable name so that we will remember that we are using transpose table for data analysis. And then we will go further for analysis. Then view the transform data that we are having. Okay, so after this, we can visualize our data with different plots. And let's see here. So PNG again, I have used to save the plot. Here I have generated box and whisker plot that we can see right now. Okay. Okay, let's generate a plot rather than opening. It will take some time. So we will try to generate. Okay, so here this is just for margin and the R code, basic R code we have. We want column 2 to column 5 data only, just to see few plots for few number of genes. So let's just see this. Here, can you see in the plot tab, we are having box plot, and for each gene, like only 4 columns we have selected. So for 4 genes, it is showing the box plot, how its expression looks in your data for the healthy samples and the disease samples. So it is showing currently the overall expression of this gene. And accordingly, you can save the box plot. You can also give more number of columns. For example, you have 14 samples, so you will give 13 here, so that because the first column we are not giving it here, that is the sample name. So we are giving all the column, and no, this is for gene actually. So we are having 100 genes, so you can use like even view box plot for 12 genes. Or let me show you just here, and I'm using column 2 to 9 so that we can see 7 box plots for 7 genes. And then for that we need, if we want to visualize here, we need 2 rows, because for 1 row it will be complicated. So in 1 row we can use 8 so that it will show at least 8 box plot. So this is just to set the margin of this area, like the box plot tab. Here we are setting the margin that I need 8 section of my plot area, and then this will show you the plots. Let's just show you here. What is figure margin is more or less. Let me, okay, because it is having less space, so we will try with 4 only and give 2 rows so that it will show you in 2 rows. Here, so now we have 8 plots and 2 rows are there, so that we can see a few number of genes and their expression level in our samples. So that is one thing in for visualization in exploratory data analysis. Similarly, you can make scatter plot and other things. So other plot like density plot and feature plot also you can make for all. It will use all the gene. So feature right now in your data, because you have transposed your data and your data is right now in transposed form for all the genes which are right now in column form or in the first row. So all the genes will be considered under the feature. Okay, so it will take more time. I will share you this one, but it is time intensive. Will take all the 100 genes. So you can try with few genes, like make a shorter table than this one. Take first few 10 genes and try to with that. But this will also work. It will take some time. So you need to wait. Don't worry that your code is not working unless it is not throwing some error. Okay. And also when it is giving error, you can just read the error carefully, try to Google it and explore. If still there is any issue, let me know. We will try to work with that. Now coming to machine learning exactly, where we are starting the splitting of data. Create a group of samples so that you will be having 60% in training set and 40% in testing set. You can increase or decrease this number also, but it is good practice that you are giving more section of your data in training part. More better the training is, better the prediction of your model will be. Okay, so sometimes we also give 70 and 25 ratio, 70 and 30 ratio. Sometimes we give 75 and 25 ratio. So that is totally depending on your data and the algorithms that you are using, or whatever you wish to try with your data. Like it is just a hit and trial method so that you can try by your own. Set seed we are keeping so that our results will be reproducible. Once we are getting one result, next time when we are trying to obtain the result, it will give the same. Okay, now we are trying to split the data. Here we have mentioned already that this will be 60 and 40 proportion of our data. So p equal to 0.60, and then training the data. So split my data, half part 60 portion of your data in training set, and in testing set split that. So okay, so we are just splitting the data here with this code. Okay, so this is done. Now we can see dimension of your data, like 19 samples total, and 101 is given, but 100 genes are there. One column is for the health sample name, and that we will remove later. So that is just the dimension of your data frame, not the training and testing set. Okay, now we can see dimension of your training and testing set here. Now we have, since distributed our data or splitted the data, so we have 12 samples out of 19 in training set and 100 genes in your data for training, and then seven samples for testing data, and then again 100 samples. Okay, so this cross validation we can do before training or after training. That's totally depend on us. So cross validation, we are giving cross validation 10. In PPT we saw cross validation 5, that how 5 times it will take for different proportion of 60% of your data, and then it will try to train your model based on your data. And then metrics, generally we take accuracy, but we are giving it here, but the model can also predict other accuracy metrics, for example specificity, recall, precision, that we saw like recently in our analysis. So just run this so that anytime during modeling we are needing this control cross validation number or the accuracy matrix, we can mention directly. So we are giving it in variable so that we don't need to use the whole code, this because this is a longer code. So we need to provide for every model this one, like either type or copy or paste, but we want to keep it short and simplified, our code readable. So we are storing it in like shorter name variable, that is control. Always use that. Remember that control we are using for cross validation. Okay, so now we will build the model using different like classifiers. So we are using k nearest neighbor first, and for that we need ISLR library or the package to first install and then load. If you are loading the library, it is giving error, it means it is not installed. So try to install that. Since my library package is already installed, so I'm just loading it. Cow plot is just to make the plot. So I will show you what plot we are generating. Cow plot actually combine two plots into one and show you there in the plot tab or in the figure form. Okay, once the package is loaded, we will try to prepare the model. Here, set seed again, we are adding because we want to run this code again and again and want to get the same results. Because a prediction model prediction may change like slightly for each modeling, like each time you are running this model based on the cross validation, because every time you are giving since you have given 10 number of cross validations, so every time it will use different set of data from your whole data that you are providing for training, and it will try to model. So that's why we always use this set.seed to get the same model. Now we will try to uh, prepare the model using the symbols column. You see, we were seeing the dimension, it was showing 101 because the first column is also included in that with the gene names, and that column is symbols column where all the list of given that healthy sample or the disease sample. It means your data is labeled. You already know that which are the healthy samples and which are the disease samples. That is used as a label for your data with respect to all the columns. So this tilted form is this expression is called tilde, and there is a dot here. That dot means it will include all the genes, all the columns after the symbol column for your this prepare the modeling. Okay, and then data will be provided for training. And what is this train? This train is given here where we decided that we 60% of our data will go in training. Okay, so training data only will be used by the model to train your model. Okay, or the algorithm to train your model. And this then TR control will be as an argument, it will give as CV control, that is for control cross validation. So we are using control here. So what we will do, we will use, let me just rename this, or we can rename it here control, because we are giving for to train the model, we want cross validation for this. Let's just train the model first. Okay, here we have trained the model and we can see, okay, no, here we have trained model warning in nominal workflow. There were missing values in resample performance measures. No, okay. So when you are using a cross validation, it will resample your data, like adding more samples or less sample, all the sample but different set. Like use the trained model but use different set of data. So that's why it is showing that there were missing values, but there were no missing values as they have already confirmed with your data. So this is just a basic warning of our data. Okay, so now we have 12 samples in our data, and I think there is, yeah. So no pre-processing was happened. Two classes there in your data, normal and the tumor samples are there. 100 predictors are there in your data. Okay. 100 predictors are there in your data. Resample was performed with tenfold cross validation. You can see the whole summary. And then there is number of tree. So, okay, this is right now what it is doing is here we have not mentioned what method we are using for this. So let me just see it here. Fit KNN. Actually, I used random forest first, so that's why we are. It is not. It is showing output for random forest. That is not for KNN. Okay, let me just add it here. Method. Where is that? Okay, so now we can see in the this this okay, description, the summary is here, and we can see the k value in the knn. So knn is k nearest neighbor. So how many nearest neighbors or how many nearest genes it has identified. Now since here you can see methods was missing here, that's why it was working with the last model that I ran for before the session. So random forest was run, that's why it was working without mentioning method. It was running the random forest only. Now we are running k nearest neighbor model, and here we can see 12 samples, total 12 samples and 100 predictors, so 100 genes, and two classes are there. No pre-processing happened because we already performed and pre-processing on our data, because no error was there, so that's why no pre-processing done. And then we have k nearest neighbors, so how many nearest genes it has like searched to make the cluster, and for that it is giving the accuracy 0.8, 0.8. So for all the, so you can also mention, like when I said the optimization of your model, there you need to mention, here also you can mention k values like k equal to 5 or k equal to 7 or 12 or 15 to improve the accuracy of your model. Okay, so that also you can try, but right now we are using the default values, and with default value it is like three set of k's, so that it can give you the accuracy. Okay, and kappa value is also for the like type of matrix, that is to see that how your model is working, but right now we are going to focus on accuracy only. And here this model is giving 0.8 accuracy, that is 80% accuracy. And we can see that it is performing well, but we want to see with other models also. So let's just see that. And summary of your model, you can see that how, what is the information, that what information it will give for k, and what is other like tuning values that you can add, other parameter values it will be given. So that information you can see with the summary values. And then feature importance, now we want to see what is the important features or important genes in our data, so that you can see with variable importance. So let's just see that here. Okay, this you can see here, the for all the genes, the importance value, it is showing that for 100, like 100 score giving for all the genes here as of now. So this does not seem a good model for us, because there is no one specific genes or few specific genes. So this is only showing 20 top genes basically, but it is showing like no variation in the importance of the genes. So as of now we will not stuck with with this model, and we'll try to explore other model. So here is the question for some of your queries, that why we use a different model for analysis, because we want to see that what other models are giving, because this model is does not seem good for us as of now, because accuracy was 80. That seems good, but we are not sure. While exploring the feature importance, it is not able to identify which gene is important and which is important for classifying the healthy samples from the disease samples. So we will not consider this as of now, or not finalize this model as of now. Let's just see the plot. So now we can see for these genes, and this is giving this score as 100. I think we are exceeding the time. Let me just quickly go through. So this is variable importance plot, and we can see with that like what is the importance of the gene. Based on this, we can also plot, make the predictions. Like now we are providing the test data to our model, and try to see how the model, this fit.knn is our prepared model based on our data, and test data that we are going to provide to our model, and see how it is predicting the results or the samples, whether it is predicting right. So in test data, we are not providing the first column, which is showing which is normal sample or which is healthy sample and the disease sample. It will itself identify whether it is a healthy sample or the disease samples. Okay, so run this prediction and positive class will always indicate normal or the healthy sample. This means that so the model itself is fixing by default that it will consider healthy sample as positive. So whenever in confusion matrix it is showing positive, so it means it is a healthy sample. Okay, so now you see the confusion matrix in the text format. You can see here, this is a tabular data, like here is the reference, that is the actual sample id, sample numbers, and these column is the predicted, and then there is a prediction that the model has selected like this norm, this healthy sample. So the reference says, or actual value says, that the model or the data have two healthy samples and total 5 disease samples. But the prediction, your model is making, your KNN model is making the prediction that there is no healthy sample. It means N indicates normal, all the healthy samples, and T indicates the tumor sample. So model is predicting that there is no healthy sample, but there are 2 plus 5, that is 7 disease samples. It means your model is not performing well or not able to predict without labels. Your data is not like model is not able to predict your data clearly, and that is not a good model for your data. So that's why you will proceed for next model. Okay, accuracy is calculated again here with the prediction matrix, like what is the accuracy of this prediction. So this is 0.71, that is 71.43%, and that is not sufficient, as we see the in confusion matrix clearly that it is not able to predict. And because maybe because of less number of data that we are using currently, or maybe because this model does not work better with small data. So yeah, again, kappa score is given here, and other p-value tests are given here, sensitivity scores, specificity score. The scores, different type of score based on accuracy matrix is given here. So balanced accuracy, you can see it is just the 50%, which is not good for our model. So this is how we actually do different type of prediction using the model, and this is used. We can also see the what is the actual labels, but we can, in confusion matrix itself, we can see what is the actual number of like number of samples in our category, and then what is the predicted by the model. Similarly, we can run support vector machine, and we can see that how it is performing on our data based on the training data that we are providing, and based on the testing data we can identify or see what is the prediction it is making based on our data, and make the confusion matrix based on that. Okay, so I think we are already exceeding with the data with this time that we are having or allotted. So what I will do, I will share you the script. You will be having some of the models code here in the script, and you can also make the like prediction with your data, and then let me know like if there is an error or any issue. We can discuss that in the next class. Okay, hope that is clear. If you have any query, just quickly drop me in the chat box. We can discuss that. Bhavani, are you there?
Yes, I'm here. Please tell me.
Okay, that's great. Actually, we are already done with the session. I was just expecting a few queries if there is any from anyone. Else, if once they will practice, they must have some query. So we can discuss in the next class. We
Okay.
can wrap it up now.
Yes, yes, yes. Before that, let's get confirmation from participants. Dear friends, is
Yeah.
that okay for you? Hope you understand today's session was very monotonous. But still, I just want to hear some live feedback. Are you okay with this? I mean, are you okay with the today's session? Please let us know. Kindly give thumbs up. Maybe the scripts and the recordings will help you on working with this. Um, Before that, I need thumbs up from all the participants if you are okay with today's session. Thank you, Abdul. Others? Thank you all for your kind feedback. Okay. As usual, we have shared the feedback link in the chat. Thank you, Mr. Abdul. Please fill it out. And if you want to give any suggestions, you can make use of that form. Yes, if you have any queries, please post it in the Telegram discussion forum. Let's meet tomorrow by the same time. Thank you all. Have a great evening.